Welcome, aspirants! Let's dive straight into today's Rungta IAS Academy Explainer. We've got an incredibly exciting and honestly highly probable topic for your GS Paper 3's Science and Technology section and for sure your personality test. Today, we're decoding a massive breakthrough, CART therapy, or what we call living drugs, and how it's being used for HIV. Let's quickly map out our syllabus for the session. We'll start with the HIV reservoir challenge. Then we'll demystify exactly what CAR-T therapy is. After that, we'll break down this new breakthrough study. And finally, we'll tie it all together with the UPSC mains and interview focus you need to fetch maximum marks. All right, Section 1, the HIV reservoir challenge. Now, you might be wondering, why are scientists globally still hunting for an elusive HIV cure? I mean, we already have pretty effective antiretroviral treatments, right? Well, let's look at why. To put this into perspective, nearly 40 million people are living with HIV globally. Now, it's true that modern medicines have turned what used to be a fast killer into a manageable chronic disease, but that's only true if patients can continuously afford these expensive drugs and strictly stick to taking them every single day. And here is the core issue. I really want you to note down this specific UPSC keyword, reservoirs. You see, the virus actually hides out in these hidden reservoirs inside the human body. If a person stops their daily treatment, the virus rebounds incredibly fast. So this hidden viral load is exactly why our current daily treatments fall short of being a true permanent cure. Moving on to Section 2, demystifying CAR-T therapy. Let's break down the core biotechnology concepts you'll absolutely need to write a top-tier answer in your GS3 paper. Take a close look at this colorized electron microscope image. The blue part is a human T cell, basically our body's immune soldier, and it's actively under attack by the HIV virus, which is shown in yellow. This shows you exactly what's happening at the cellular level, and it perfectly illustrates why we need to supercharge these specific immune defenders. And to do exactly that, scientists are turning to living drugs. Simply put, these are genetically engineered immune cells that are infused back into a patient's body to fight disease. Now, remember, this powerful technique was originally developed to cure certain types of cancer, but researchers are now boldly tweaking it to target HIV, a real game changer. So how does the CAR-T therapy mechanism actually work? Well, it's a clear three-step process. Step 1, extract those immune soldiers, the T cells, from a person's blood. Step 2, genetically engineer them in a lab into these supercharged living drugs. And Step 3, infuse them right back into the patient. Aspirants, if you can draw this exact flowchart in your mains answer sheets, it will definitely fetch you extra marks. Now, for HIV specifically, scientists at a nonprofit called Caring Cross did something brilliant. They gave these CAR-T cells dual features. First, they programmed them to be much better at finding and killing HIV-infected cells. Second, and this is the genius part, they engineered them with built-in protection against the HIV virus itself. With this added armor, these cells can reproduce enough to keep HIV in check without being destroyed in the process. Great, on to Section 3, the breakthrough study. Let's analyze the hard clinical evidence from a recent early-stage experiment that was presented at the American Society of Gene and Cell Therapy. This study tested different dosing strategies on people who actually stopped taking their normal medication the very day they received their CAR-T cells. 6 patients got a small amount of mild chemotherapy just to clear some space for the new T cells. The outcomes? Literally remarkable. 2 strong responders saw their HIV drop to undetectable levels. One suppressed the virus for nearly a year, and another for almost 2 whole years. And the crucial point here, they accomplished this entirely without needing their usual daily HIV medicines. Dr. Stephen Deeks, who led this research, summed it up perfectly. He said there is a real need for a one-and-done, safe and scalable cure. And this is one of the strategies that we're pursuing. I want you to underline that phrase in your notes, one and done, safe and scalable cure. That right there is the ultimate socioeconomic goal for global public health, tying directly into your GS Paper 2 health syllabus. Finally, Section 4, UPSC mains and interview focus. Let's consolidate everything we've learned with the critical analysis and the limitations that an IAS aspirant simply must demonstrate. A top-tier mains answer has to be balanced, so you must acknowledge the caveats. First, as researchers cautioned, this is an early-stage experiment. We still need much larger and longer studies. Second, patient history is a huge factor.
The successful responders had started their original HIV treatment very soon after getting infected. This makes sense because people treated early tend to have less HIV hiding in those reservoirs and a generally healthier immune system. Finally, contrast this with past attempts. A handful of patients were technically cured via stem cell transplants for cancer, but that procedure proved way too risky for the general population.
To wrap up our masterclass, I want to leave you with a classic, thought-provoking UPSC interview panel question. Is a highly personalized, genetically engineered living drug a viable public health solution for the developing world? As an aspirant, you've got to weigh this cutting-edge science against the harsh economic realities of healthcare infrastructure in countries like ours. Think about it, structure your arguments well, and keep studying hard. Thanks for joining this explainer.